Now we are going to discuss about the food poisoning in detail. So this food poisoning question is always asked in a clinical scenario based question. So they will give you a clinical scenario that a number of people or a group of people are coming to your emergency with the complaints of vomiting, diarrhea, and the abdominal cramps, etc. And on history taking, you got to know that have been in the same they had been in the same marriage party or in the same birthday party, and from there they are coming with all these complaints. So how are you going to approach this case? So in that case you have to diagnose it as a case of food poisoning and then answer all the questions sequentially. So first of all, they may ask you what are the causative organisms in that particular clinical scenario. So I have mentioned all the causative organisms of the food poisoning here in a tabular manner. So if they give you a clinical scenario that they are coming with all those clinical features within 6 hours of eating the food, then you have to answer the causative organism as Staph aureus or the Bacillus cereus, because these two are concerned with causing the food poisoning within 6 hours of eating the food. Then if they give you the clinical scenario that the group of people have eaten the food in the night and then they are coming in the morning to your emergency, that means there is a time gap of 8 to 16 hours from eating the food till the presentation of the symptoms. Then you have to answer the causative organism as the Clostridium perfringens and the Bacillus cereus, because these two are concerned with causing the food poisoning in these time frame after eating the food. Okay. And if they give you a clinical history that there has been a time gap of more than 16 hours from eating the food till the presentation of the symptoms, then you have to mention these organisms. Okay, what are those are the Vibrio cholerae, the enterotoxigenic E. coli, the enterohemorrhagic E. coli, the non-typhoidal salmonella. What are the non-typhoidal salmonella? Those are the typhoid type Salmonella typhimurium, the Salmonella enteritidis. So those are all the non-typhoidal salmonella. They cause the food poisoning and all. Okay, so you have to mention the non-typhoidal salmonella. Then you have to mention Shigella and the Vibrio parahemolyticus, because they are causing the food poisoning after 16 hours of eating the food. Okay. So based on the clinical scenario, based on the time gap from eating the food till the presentation of the symptoms, then based on that time gap you have to answer the causative organism. But suppose that they did not mention the time gap. Okay, they did not mention the time gap. Then what will you do? You have to just make a table like this. Okay, you have to just make a table like this. You make a make a table of 1 to 6 hours incubation period, then a table of 8 to 16 hours, and then a column of more than 16 hours, and then write all those organisms. If they did not mention the time gap from the food eaten till the presentation of the symptoms, so you have to make this table then. Okay. And after answering the causative organisms, then you have to diagnose the case. Okay, so diagnosis of the food poisoning is not only based upon the laboratory diagnosis, because there are so many organisms which can cause the food poisoning. So you have to narrow down to a particular group of microorganisms which you will focus in your lab to diagnose the particular microorganism causing the food poisoning. For that you have to start from the beginning, and by beginning I mean that you have to start with the meticulous history taking. So what does that meticulous history taking help, or how does that meticulous history taking help? This meticulous history taking will help you in narrow down your diagnosis. Okay, you narrow down your group of organisms to which you will focus more in your laboratory diagnosis part. Okay. So in the meticulous history taking, first of all you have to ask about the time gap from the food eaten till the presentation of the symptom to each of the patients. So you have to ask whether the symptoms has started within 6 hours of eating the food, or the symptoms has started after 8 hours but within 16 hours of eating the food, or the symptoms has started after 16 hours of eating the food. Because in all of these situations the organisms will vary. Okay. And based on this time duration we will narrow down to a particular microorganisms as well. So this These asking this onset I mean asking this incubation period is very important. Then you have to ask what type of food they have consumed, because again this will also help us in narrow down to a particular microorganism. How will it do so? So let's see here that if the patients say that they have eaten dairy products and pastries, then your suspicion will go towards the staph aureus. Your suspicion will go to the staph aureus. Then if they say that they have eaten the Chinese fried rice, then your suspicion will go to the bacillus cereus. If they say that they have eaten some seafood like prawn or shellfish, then your suspicion will go to the vibrio cholerae or the vibrio parahemolyticus as a causative agent of the food poisoning in those patients. If they say that they have eaten the eggs or meat, then your suspicion will go to the non-typhoidal salmonella or shigella as a causative agent of their food poisoning. If they say that they have eaten the ground beef or raw vegetables, then your suspicion will go to the enterohemorrhagic E. coli. If they say that they have eaten beef or poultry, then your suspicion will go to the Clostridium perfringens. So, in this way, this all the type of food that they have consumed will help us in narrow down to a particular microorganism and will help further in the laboratory diagnosis part. Then we will also ask for the symptoms, okay, because the symptoms are also important to narrow down. Because watery diarrhea, we see that the watery diarrhea is mostly seen in the enterotoxigenic E. coli infection or the Clostridium perfringens infection, while rice water stool is world famous and it is seen in case of the Vibrio cholerae. This will help us in narrow down to the Vibrio cholerae. Then if the patient say that there is vomiting, diarrhea and abdominal pain, all of these are occurring to that patient, then we can consider the staph aureus or the bacillus cereus as the causative agent, okay. If there is occurring bloody diarrhea, then we may think of the enterohemorrhagic E. coli, Shigella, and the Vibrio parahemolytica. So, in this way, the symptoms are also helping us in and these symptoms are also helping us in narrowing down to the particular microorganism. So, hence this meticulous history taking is very important. So in the meticulous history taking, we have to first ask the time gap between the food taken till the presentation of the symptoms, that is the incubation period. Then we have to ask the what type of food that they have eaten, and then we have to ask about the symptoms, like what type of diarrhea is occurring, whether there is vomiting along with diarrhea or vomiting without diarrhea, or there is abdominal pain associated or not, or there is fever or not. So these all symptoms will be helping us in narrowing down. So after doing all this, you have to go to the lab diagnosis part. So in the lab diagnosis, we'll first start with the sample collection, and here as the symptoms are vomiting and diarrhea, so obviously we will be collecting the vomitus and the feces for sure. But one more thing we have to collect here is the food material, because if a large number of people from a community are affected by going to a particular marriage or a particular the birthday party, then we have to check that food material that is supplied to that marriage party or the birthday party, so that we can get to know about the source of infection, okay. So for that we have to send some uh, administrative people to go there and get some food particles or raw vegetables from those parties, and check it under in the microbiology labs to identify the particular microorganism causing the infection to a large number of people of the community. So that's why we have to collect the food materials also. And the food materials can be the cooked food or maybe a raw foods. So the cooked foods, how do we preserve those cooked foods? So cooked foods, we will take 10 gram of that food and will homogenize in 90 ml of sterile water. So this is for cooked food. And what if there is raw vegetable or fruits? So in the case of the raw vegetables or fruits, we will take 100 gram of that food and will mix it in 100 ml of sterile water and will shake it well. And after shaking it well, we will get the organisms dissolved in the water. So these are the two ways by which we will process the food materials, okay. If it is a cooked food, then we have to homogenize it in 90 ml of water. If it is a raw food or some kind of fruit is there, then we have to just shake it well in the sterile water and will preserve that dissolve, I mean will preserve that water in which the food has been shaken off. After that we have to transport the specimen. Okay, so how are we going to transport the specimen? Of course we know that the specimen should be transported immediately to the labs, because the microorganisms may not be viable in some drastic conditions, weather conditions of India. So hence the transport media are very important. And how are we going to select those transport medias? That is basically based upon the meticulous history taking, because that history will help us to select a particular type of transport media. Like if from the history we get to know that the patient might be suffering from cholera, then of course we will be using autoclaved seawater or carry layer medium for transporting of the specimen. Okay, if we suspect from the history taking that the patient might be suffering from salmonella or shigella, then we will use the buffered glycerol saline for transport of the specimen. If we suspect that patient may be suffering from Clostridium perfringens, then we will use the Robertson cooked meat broth for the transport of a specimen, isn't it? So that's where the history taking comes into play in our diagnosis part. Okay, hence that history taking is very important in the medical profession, and you should be very clear and thorough with the history taking. You will get to know about this when you come to your final years. Okay, let's come to the next point, that is the food processing. So after the processing, after we have homogenized that food particles, then we have to serially dilute it, serially dilute and dilute it, and then we have to filter it. Okay, why do we want to filter it? We want to filter because we do not want the food particles. No, we want just the bacteria to pass through that filter. So we want to separate that food particles after homogenization. So by filtration the food particles will get separated and the bacteria will get filtered into the filtrate, and that may be used for the culture and all. So that is the use of the food processing, I mean that is the importance of the food processing. And after that we have to do the use the enrichment broth. We have to use the enrichment broth before culture, because we know that in the feces sample or in the stool sample, uh, we there is generally commensals also in the stool samples from the gut of the human being, because the human gut contains a large number of commensals. So that's why mm we want to grow the particular type of our target organism. Okay, we do not want our commensals to grow in the culture media. For that we have to use these enrichment broth, because this enrichment broth will help us in selectively growing the target organism. So that's why enrichment broth is also important in the diagnosis part. Okay, to grow the target organism. And then after that we will do the culture. So how are we going to do the culture? So for the culture we will use the filtrate, and the specimen from the enrichment broth plus the vomitus or the stool can be inoculated into any one of the culture media. So culture media that we are going to use are the McConkey agar, the blood agar, and the Robertson cooked meat broth. After inoculation, then we have to incubate it at 37 degrees centigrade for 24 to 48 hours. And after incubation we'll see the colony characteristics. So the colony characteristic that we see in the McConkey agar, there may be lactose fermentation or there may be non-lactose fermenting pale colonies. So if there is lactose fermentation, that means if we are seeing the pink colonies out there on the McConkey agar, we may think of there is E. coli infection. That E. coli may be enterohemorrhagic E. coli or the enterotoxigenic E. coli. Okay, and if there is non lactose fermenting pale colonies, then we may think of the salmonella, that is the non typhoidal salmonella, and then Shigella or the Vibrio cholerae. Remember that Vibrio cholerae is a late lactose fermenter. If we leave Vibrio cholerae for some more duration, then we will start fermenting that lactose, but in that selected amount of duration it will not ferment. Okay, it is just like Shigella soni. Shigella soni is also a late lactose fermenter. Okay, so uh, this this is how the McConkey agar will help us in separating these in differentiating between these two organisms, that is lactose fermenting and the non lactose fermenting organisms, and narrow down to a particular organism. Then blood agar. So the characteristic features that we see in the blood agar may help us in reach to a particular organisms also, because if we see that there is narrow zone beta hemolysis, then we may think of the staph aureus infection. If there we see the target hemolysis, then we may think of the clostridium perfringens, because target hemolysis is seen in clostridium perfringens by the toxins released by the clostridium perfringens cause this target hemolysis in the blood agar. If we see that there is irregularly spreading beta hemolytic colonies, then we may think of the bacillus cereus. Okay, so this is how the colony characteristics on the blood agar may also help us in differentiating between organisms. Then the rcm media, if in the rcm media we see that the meat particles have turned pink, then we may think of the clostridium perfringens infection. Okay, so this is how the colony characteristic is helping us in narrowing down to a particular microorganism. Okay, now one important mcq question that may be asked in the university exams is that what is the selective media of bacillus cereus. So please remember the selective media for the bacillus cereus are the MYPA and PEMBA. So MYPA pemba is the selective media for the Bacillus cereus. Please remember this. This may be asked in the university exams as a MCQ, that the selective media for Bacillus cereus is MYPA pemba. What is the selective media for Bacillus cereus? It is MYPA and pemba. Now let's come to the our topic. So after seeing the colony characteristics, then we have to do the culture smear, and we have to see the organisms under the microscope after the gram staining. So with the discrete colonies on the colonies with the discrete colonies on the culture medias, we will make a smear and will gram stain the smear. After gram staining, if we are seeing that there is gram positive cocci in clusters, then we may think that it may be staph aureus. If we see that there is gram positive bacilli with spores in short chains, then we may think of bacillus cereus. If we see that there is gram positive bacilli with spore which is arranged singly, then we may think of clostridium perfringens. If we see that there is gram negative rods arranged haphazardly, then we may think of the enterohemorrhagic E. coli, the enterotoxigenic E. coli, or the Shigella. If we see that there is gram negative comma sepet bacilli, then we may think of vibrio cholerae. If we see that there is gram negative bacilli with bipolar staining, that means that is there is safety pin appearance. If we see under the microscope, then we may think of vibrio parahemolyticus as the causative organism. So this is how we have narrowed down to different types of microorganisms based on the gram staining. Now for confirmation, we will have to do the biochemical tests. Okay, so identification part is done is started with the catalase and the coagulase test. If both the catalase and coagulase is coming out to be positive, then we may think of staph aureus. And for we'll also have to do some other tests like the catalase, oxidase, citrate, urease, and the indole test. So if the catalase test is coming out to be positive and indole oxidase negative, citrate negative, urease negative, and indole again positive, then we have to think of E. coli. It may be EHEC or ETEC as the causative as organism of the food poisoning. If we are seeing that only the catalase is positive and rest all other tests are negative, that means it can be salmonella. Then if we see that again Shigella, that the catalase is positive but all other tests are negative, that means it can be Shigella. Then we have to see the vibrio cholerae. So in the vibrio cholerae, if you see that the catalase oxidase and the indole are positive, but the urease is negative and citrate may be positive or negative, then we may think of the vibrio cholerae here. Okay, so by this we will narrow down or will come to the diagnosis. Okay, that what is the organism which has caused this food poisoning. Now this is very fascinating to see and write, but how are you going to remember this? So for remembering, you have to remember the Kokui score. Remember the Kokui score of the different bacilli that I am talking from, that I have talked in many lectures of mine. So you must have been knowing it if you are following my lectures. So the Kokoi score is COCUI. The COCOI scores of different bacilli is written here. Okay, like for E. coli, the COCOI score is PN3P. For Salmonella, the COCOI score is PN4. For Shigella, PN4. For Vibrio cholerae, P2VNP. Okay, so COCOI means catalase, oxidase, citrate, urease, and indole. So PN3P means the catalase is positive, then oxidase, citrate, and urease are negative, so N3, and then indole is again positive, so P. So that is the meaning of the P and 3P. Okay, that is the meaning of the COCOI score P and 3P in case of E. coli. So see here the COCOI score in the case of E. coli is P and 3P. So see here catalase is positive, indole is positive, rest all other are negative. So COCOI score of E. coli is PN3P. Similarly, for Salmonella, it is PN4, for Shigella, PN4, and for Vibrio cholerae, it is P2VNP. Okay, so this is the biochemical test that we have to do for diagnosing the food poisoning. Okay, after that we can also do the direct toxin detection, like many of the organisms cause the food poisoning by their toxins, like the our bacillus cereus, like our enterotoxigenic E. coli, like our Shigella. So they may cause this cause this food poisoning with their toxins. So in those cases we have to detect the toxin. If we detect the toxin, then the food poisoning itself will be diagnosed. Okay, so the toxin can be detected in the stool specimen by ELISA or by the latex agglutination test. So with the help of the ELISA and the latex agglutination test, we can detect the toxins, the heat stable and the heat level toxins of the enterotoxigenic E. coli, or the toxins of the Bacillus cereus, or the toxins of the Shigella. Okay, so those all toxins will be detected by this ELISA or the latex agglutination test, and that will help us in diagnosing the food poisoning as well, plus the causative organism of that food poisoning as well. Then we have molecular methods, of course. So with the molecular methods, we will directly detect the genes which are producing those toxins, and once we detect the genes, the organisms will itself be demonstrated. Okay, so PCR can be used for detecting the genes which are producing those toxins. Okay, other than that we have and after all that we have antimicrobial susceptibility testing. So with this we will get to know about the based antimicrobial out there for that particular type of food poisoning. So this is the whole the sequence in which we have to diagnose a case of food poisoning if it is presenting to a emergency. Okay, so you have to start with the meticulous history taking. You have to take the history of the you have to take the history of the duration from the food taken till the presentation of the symptoms. That time gap should be asked for to the patients, and then you have to ask for the what type of food that they have consumed. Okay, what type of food that they have been consumed, that they have consumed. And after seeing the food consumed, then you have to ask for the symptoms, what type of symptoms they are facing. Then you have to start with the laboratory diagnosis. In the laboratory diagnosis, you have to collect the specimen, not only the vomitus and the flesh tissue, but also the food material. And you have to homogenize the food materials like in this way. And then you with the raw foods or the fruits or the raw vegetables, you have to shake it well. Okay, and then you have to transport the specimen based on your history taking, based on you know the history, what type of organisms you are suspecting. Based on that only you have to select the transport media and transport the specimen. And then you have to do the food processing, because you have homogenized the food, so you have to separate the food particles, so that the filtrate can be used for culture media na. So that's why you have to do the food processing. After that, in case of stool samples, you have to use the enrichment broths to selectively grow the target organism. So enrichment growth is important. And after that you have to do the culture. For culture you have different types of medias. And after culture, then you have to see the colony characteristics on different culture media. Plus you have to remember this selective media of the bacillus cereus. And also then you have to see the culture smear and the gram staining characteristic of the different bacteria that will help you in narrow down to a particular microorganism. After the gram stain, then you have to do the identification part. That is, the identification part include these catalase, oxidase, citrate, etc. tests that you have to do, and write the test results of all the suspected microorganisms, and then you have to detect the toxins with the help of ELISA or the latex agglutination test that you can use. Other than that, you have got the PCR. By the PCR, that is the molecular methods of detection. By the PCR, you can detect those toxins. And then antimicrobial susceptibility is also tested. I mean, antimicrobial susceptibility testing can also be done to find the base antimicrobial for the food poisoning. So that's all for the food poisoning. I hope I have been able to make you understand the whole approach of diagnosing the food poisoning, and from now you will not face any difficulty in the food poisoning question. That all that's all for the food poisoning.